This is part 3 of the chapter 2 lecture notes on prokaryotes. Let me get it set up. We left off on this page, and I want to come back to it because I didn't quite finish it. All right, so let's just go back and review. So up until now, basically in Bio 111, we learned about chemoheterotrophs like animals and fungi and some protozoans. And we learned about photoautotrophs. So we learned about chemoheterotrophs, example being animals and fungi. And then we learned about photosynthetic organisms, which are photoautotrophs. So plants would be an example of a photoautotroph. So examples of photoautotrophs are plants. Examples of chemoheterotrophs are humans. Now, what's different from what we've learned before is that bacteria can also be photoheterotrophs or chemoautotrophs. So, to decide if it's an autotroph or a heterotroph, you ask where the energy comes from. If the energy, I said that wrong. Let me start over. To ask whether it's an autotroph or a heterotroph, you ask where the carbon comes from. If the carbon comes from elsewhere, and elsewhere means food, if the carbon is obtained from food, from consuming food, consuming some type of inorganic molecules for food, then it's a heterotroph. If the energy comes from light, then it's a photoheterotroph. Okay, so there are some prokaryotes that's how they obtain energy, is they're photoheterotrophs. They consume food, but they still get their energy from sunlight or ultraviolet light. And then if they're chemoautotrophs, that tells you they make their own food, but their energy source is breaking down some sort of chemical. So their energy source is their carbon source is carbon dioxide from the atmosphere. And their energy source is that they break down some sort of inorganic chemical. They oxidize it or break it down. Then we have the relationship between prokaryotes and the carbon cycle. It says prokaryotes play a significant role in continuously moving carbon through the biosphere. Primarily, prokaryotes are a lot of bacteria, particularly, are decomposers. And when they decompose dead organic molecules, they release carbon dioxide into the atmosphere. And so that's the main thing. The main way that they release carbon dioxide into the atmosphere is the same way we do, by releasing it through respiration and also by decomposing other organic molecules, dead organic material. In the nitrogen cycle, prokaryotes also can play a key role. There's decomposers again, okay? They're still decomposers, and whenever they decompose organic chemicals that contain nitrogen, they release the nitrogen into the atmosphere. There are bacteria that are called denitrifying bacteria that release nitrogen into the atmosphere. Decomposers don't release nitrogen into the atmosphere. They release nitrogen in the form of ammonium. And nitrogen-fixing bacteria take nitrogen from the atmosphere, which is unusable to most living things, and convert it into a usable form. Nitrifying bacteria convert ammonium produced by the nitrogen-fixing soil bacteria into nitrites. And nitrites are more of a usable form of nitrogen. Well, that's the reason we use nitrogen for fertilizer. So denitrifying is taking the nitrites and converting them back. That's a kind of a backwards reaction. These are bacteria that convert nitrates back into nitrogen, the nitrogen in the atmosphere. which is an unusable form of nitrogen. I don't know if you remember, but about 80% of the atmospheric gases are nitrogen gas. Mostly the rest is oxygen and other gases. So then we have bacteria that act as decomposers, which break down the nitrogen compounds in plants and any dead organic material, and convert it to ammonium. And then we have soil bacteria that fix nitrogen. They actually take nitrogen from the atmosphere and convert it into ammonium. Then we have nitrogen fixing bacteria that literally live in the root nodules of legume plants. Soybeans would be an example of nitrogen fixing bacteria that have a symbiotic relationship with the soybean plant. Okay, so those also convert atmospheric nitrogen into ammonium. And then we have nitrifying bacteria that can take the ammonium and convert it into nitrites, which is usable for other living things, usable as kind of like fertilizer for plants, and then animals eat the plants, and that's kind of how that works. Okay, so prokaryotes do not always cause disease. Many prokaryotes are beneficial, as we saw with the decomposing prokaryotes and the prokaryotes that contribute to the nitrogen cycle, the nitrogen fixing ones. But they can cause disease. They can cause disease. So we're going to talk about those. If the disease is an epidemic, that definition is that the disease occurs in an unusually high number of individuals in a population at the same time. Remember, a population is just members of the same species living in the same area. If it's pandemic, it's widespread, usually worldwide. And then if it's endemic, it's constantly present, usually at low incidence in a population. Okay, so they don't give examples. All right, so let's see. I'm not absolutely for sure, but I can tell you for viruses, everybody knows, like for viruses, HIV is pandemic. It's a worldwide epidemic. Or it definitely was in the 80s and the 90s, and I think it might still be classified like that. But bacterial diseases that are widespread worldwide, that would be a pandemic. It would be an epidemic, we've had incidences, epidemics of bacterial infection, MRSA. I know that's more of an endemic now, although it's harder to treat than a regular staph infection. It's an antibiotic resistant staph infection, but we've had it's so great if they had put some examples in the notes. But anyway, let's see. I can only think for epidemic, I can only think of like the really bad ones, but there's the bacteria, the flesh eating, bacteria that caused an epidemic several years ago. I can't remember where, that's the bad thing. But endemic is going to be bacterial infections that, cause things like ear infection, sinus infection. That's constantly constantly present in a population. All right, so in the reading in your study plan is going to give you a lot more examples, and I encourage you to spend some time on examples of disease-causing bacteria. Biofilms, by their nature, are resistant to attempts to destroy them. And we talked about what a biofilm is. It's just a lot of bacteria held together by a film, of polysaccharides. Biofilm infections develop gradually. Sometimes they do not cause symptoms immediately, but they are rarely resolved by host defense mechanisms such as our immune system. Once an infection is established, it's very difficult to eradicate it because biofilms do tend to be resistant to most antibiotics. So if an infection grows to the level of a biofilm, if it becomes that complex, then it's really difficult to eradicate it. That's when, like, if it were in a human, they would have to do probably some tissue removal. Okay, so foodborne illnesses or food poisoning comes from the consumption of pathogenic bacteria, viruses, or other parasites that contaminate food. They can also be protozoans, which are eukaryotes. In the past, it was relatively common to hear about botulism, but not so much now because botulism comes from the, Clostridium botulinum. Finally, I can give you some examples. Clostridium botulinum bacterium, and that it comes from unsafe canning practices, and people don't really can their own food as much anymore. So that's why you don't hear about botulism. Most modern cases of food poisoning are linked to produce. For example, E. coli, which I can write that one out for you too, Escherichia coli. And remember, these are scientific names, so we should technically underline them or write them in italics. Okay, so superbugs, they're not really bugs, they're bacterial pathogens that are resistant to traditional antibiotics. And with viruses, we use vaccines to prevent. With bacterial infections, we use antibiotics to kill or to stop the growth of. So it's a chemical produced either by microbes or synthetically by man that is hostile to the growth of other organisms, particularly bacteria. Antibiotics particularly are used to treat bacterial infections. Today's news and media often address concerns about an antibiotic crisis, antibiotic resistant crisis. So the main causes of resistance is the abuse of antibiotics in the past. The imprudent and excessive use of antibiotics has resulted in natural selection, selecting bacteria that contain a resistant gene, so resistant forms of bacteria. And so the antibiotic will kill the infecting bacteria and only the resistant forms remain. And if you use if you overuse the antibiotic, there's more chance of the resistant bacteria remaining and becoming the dominant strain. So that's the reason you can't automatically go to the doctor and get antibiotics anymore. But I can remember when I was a child, and I don't mind telling you, I'm 47. So there's a lot of things different when I was a child. But when I was a child, when you were sick, when you had a cold or anything, you went to the doctor, they gave you penicillin. They didn't even really think about it much. Penicillin is practically, ineffective now. I mean, because of that. So, all right. So how does resistance develop? First, you have to have lots of germs, lots of bacteria. And just a few of those are drug resistant. And the drug resistants are the ones in purple. So you've got a whole lot of bacteria with just a few that are resistant to the drugs. And if you don't overuse antibiotics, then antibiotics are effective. But anyway, the second stage is the antibiotics kill the bacteria causing the illness. And they also can kill good bacteria. So you're left with the ones that are resistant. And those that are resistant reproduce. The drug-resistant bacteria now are allowed to grow and take over. And it says some bacteria even give their drug resistance to other bacteria through conjugation or transformation or even transduction. Their drug resistance is in the form of that little plasmid. So they can actually give their resistance to other bacteria, creating what we call, this process is creating what we call superbugs.